Assalamu alaikum. So this is the second lecture of biochemistry in the hematopoietic lymphatic system, and in this lecture we will continue talking about hemoglobin, and here we'll talk about regulation of hemoglobin function and the different allosteric effectors that regulate the switching between the T state of hemoglobin and the R state of the molecule. So again, the main resource for this lecture is the lecture itself, whatever I say. You can also go to any other resource if you wish that provides the same type of information. Now, probably Mark's Basic Medical Biochemistry textbook is an excellent resource. The thing is, topics related to hemoglobin, things that we've covered in the first lecture and in this lecture, and as well as in the following lecture, you can find them in the book sporadically. So they exist in different pages, different sections. So the best thing that you can do is go to the index at the end of the textbook and check for hemoglobin, the different topics on hemoglobin, and you can go to the pages and refer to them. Now, probably chapter 7 contains most of what I will be talking about in this lecture right here. These are known as allosteric effectors. Now, the idea is that a hemoglobin molecule, the hemoglobin is an allosteric protein, meaning that it goes through different structures, R and T. And these two different structures are regulated by binding to different proteins or different molecules, I'm sorry. These different molecules can bind to the molecule and they can change its structure from, the active form or the high affinity form to the low affinity form. Now, these molecules do not need to bind to the same place where the substrate, quote-unquote substrate, or the original molecule like oxygen, for example, bind to. So they can bind to regulatory regions other than if we're talking about an enzyme, the active site. Okay. So in case of hemoglobin, they bind at places other than the oxygen binding site. Okay. Now, these are the effectors that we will discuss in this lecture. We'll talk about hydrogen ions, we'll talk about carbon dioxide, 2,3-bisphosphoglycerate, chloride ions, and carbon monoxide. And these are all known as heterotropic effectors. So oxygen is a homotropic effector because binding of an oxygen would affect binding of another oxygen molecule. So we're talking about the same molecule. So here we're talking about binding of these molecules would affect binding of a different molecule, which is oxygen in this case. So these are known as heterotropic effectors. So let's start talking about the hydrogen ions and what they do. So hydrogen ions are basically, we're talking about pH. And the idea here is that as pH goes down, affinity goes down as well. So the more hydrogen ions exist surrounding hemoglobin, the lower the affinity is. So you can see in here, this is at 7.4, that's physiological pH, so it looks like this, that's in green. So if you decrease the pH, what will happen is that the affinity decreases. And this is measured by P50, right? So the idea is that you would need more oxygen to get to 50% saturation of the hemoglobin molecule or hemoglobin molecules. Similarly, if you increase pH, that is, if you lower the concentration of protons, affinity increases. And this is known as the Bohr effect. That's after a scientist known as Bohr. So this is known as, so the effect of pH on oxygen affinity in hemoglobin is known as the Bohr effect. Same thing here. So what do protons do exactly? Now, changing the pH results in changing the protonation state of a number of amino acid residues within the hemoglobin molecule. Now, I'm not going to talk about all of them, but I'll focus on one of them, and that is histidine 146. Now, histidine 146 of the beta subunit is the very last amino acid residue. And so it has two charged groups. You have it has the carboxyl group, the carboxy terminus, that is, of the beta subunit, and the R group of histidine. And these two groups can form electrostatic interactions. Now, the carboxyl group forms electrostatic interactions with lysine that exists on the alpha subunit. Okay? And the second one is between the R group of histidine 146 with aspartate 94. Okay? So what happens here is that whenever you have switching between the T state to the R state, that results in changing the pKa of histidine 146, the R group of the histidine 146. It goes down from 7.7 to 7.3. So what is pKa? pKa is the pH where the concentration of conjugate base is equal to the concentration of the acid itself. Okay, that is the unprotonated over the protonated. Okay, so reduction in pKa means that the R group, or it means that the histidine would lose the proton much easier. Okay, so when the R group loses the proton, there is a loss of the positive charge, and as a result there is no more interaction between the R group of histidine with aspartate. So the electrostatic interaction breaks up. Okay, and the molecule becomes more relaxed, that is in the R state. Now the question is, where do protons come from? So in tissues you have metabolism and you have the production of CO2, that's in Krebs cycle. So CO2 combines with water, carbonic anhydrase, forming the carbonic acid, which dissociates into the bicarbonate ion and protons. So the pH in tissues is lower than it is in the lungs. That's a result of metabolism, production of a lot of protons. So that would cause the protonation of histidine 146 and the formation of the T form. And that makes sense because now in tissues what you need is to have hemoglobin in the T state, in the low affinity state, so that oxygen is released in tissues. Okay? So we said that protons come from metabolism, the formation, generation of carbon dioxide in tissues, and that results in the formation of protons. So that's the first mechanism. Okay, so the idea here is that the more CO2 you have, the lower the pH. So that's pH of 7.6 at 25 torr of CO2. This is 7.4 when the pressure of carbon dioxide is 40 torr. When it is 60 torr, pH is now lower, it's 7.2. So that's the first mechanism by which CO2 can switch the hemoglobin from the R state to the T state in tissues. Now the second mechanism is formation of carbamates. So what happens is that CO2 itself can bind to the free alpha amino terminal groups of polypeptides, forming what is known as a carbamate. And this would create the formation of more negative charges. That is, you have more electrostatic interactions that can take place, and this stabilizes the T state of the hemoglobin molecule. So we have two mechanisms, right? The first mechanism is that CO2 can form hydrogen ions, lowering pH, and the second mechanism is that CO2 can bind directly to the hemoglobin molecule, stabilizing the T state. The question is, which one is more important than the other. Now this was done in an experiment where pH is stabilized using buffers. Remember, we talked about buffers before. So pH can be stabilized, but CO2 is not. So in here you have three samples. You have a sample where pH is 7.4, no CO2. And here you have pH of 7.2, no CO2. So right here between red and blue, what you're doing is that you're comparing the effect of pH, because that's the only variable. That's the only thing that is different. It's hydrogen ions, not CO2. And you can see, compare red versus blue, you can see that there's a shift in the saturation curve to the right, meaning that there is lower affinity when the pH is lower. Okay, that is, again, you need more oxygen to fill 50% of the hemoglobin molecule, to saturate 50% of hemoglobin molecules. Now, in the third sample, what we've done here is that pH is 7.2, but there is CO2. So you compare blue to purple. pH is the same. The only thing that is different is the concentration of CO2. And as you can see here, there's again shift to the right. So CO2 lowers affinity as well. And it turns out that 70% of the effect of CO2 is a result of forming protons. And 25% of the effect of CO2 is due to the formation of the carbamates, or what is known as the carbamino compounds. So CO2 is transported. How does it get transported? Well, it's transported as a bicarbonate ion predominantly. So 60% of CO2 is transported in blood in the form of the bicarbonate ion. So what happens is that CO2 leaves tissues, it goes into red blood cells. In red blood cells, some CO2 is bound to hemoglobin, but the majority is converted to the ions, to the bicarbonate ion and protons, and that's how, and bicarbonate ion goes out, and that's how CO2 is transported. So about 30% of CO2 is transported as a bound form to hemoglobin, and very small percentage of CO2 is transported in the form of dissolved gas. Now, remember, what happens here is that you have always formation of protons right here. And even though, and this is known as but even though there is formation of protons, there's no change in pH overall. And this is known as the isohydric shift, which is a result of hemoglobin acting as a buffer. Remember, we talked about the bicarbonate ion, and we said that it's the predominant buffering system in blood, but also hemoglobin plays a very important role whereby it can bind to protons. It can absorb protons. So hemoglobin plays a very important role in maintaining blood pH, a phenomenon known as isohydric shift. So let's talk about the other allosteric effectors. So there is a phenomenon known as the chloride shift. And this is an important phenomenon. So what happens here is that you have this transport of the bicarbonate ion in and out of red blood cells. That means that there is charge movement across the plasma membrane, and the difference in charge must be maintained. So what happens is that whenever you have exit of the bicarbonate ion out of red blood cells, this is balanced by entry of chloride ions. And whenever the bicarbonate ion gets into cells, the chloride ions would go out. So this is known as the chloride shift, and it maintains charge difference across the plasma membrane of red blood cells. So again, what happens here is that in tissues you have production of CO2, CO2 goes into red blood cells and it combines with water bicarbonate anhydrase, forming it dissociates into the ions, bicarbonate ions and protons. Now the bicarbonate ion goes out and the chloride ions would get in, again balancing the charge. Now here you have hemoglobin mainly in the T state. Part of it is that you have the Bohr effect, right? So you have the transition of hemoglobin from the R state to the T state, and that facilitates the dissociation of oxygen from hemoglobin, and oxygen gets into tissues. Now the opposite is true in lungs. So in alveoli, what happens is that the bicarbonate ion goes in, chloride ions would go out. Okay. Now again, bicarbonate ion forms CO2 and CO2 is exhaled. At the same time, you have hemoglobin being in the R form, so it can, with high affinity state, it can form, it can complex or it can bind to oxygen atoms. So, question is there an effect for chloride on the transition of hemoglobin from the T to the R state and vice versa? And the answer is yes. So here's what happens. This is what chloride ions do. Once they increase in level inside cells, they form electrostatic interactions with the N-terminus of the alpha-2 chain, and chloride ions can also form electrostatic interactions with the R chain of arginine 141 of the alpha-1 chain. This arginine is the very last amino acid in the primary structure of the alpha-1 chain. Now remember that the alpha-1 chains are composed of 141 amino acid residues. So that increases the number of electrostatic interactions within the hemoglobin molecule, and that would stabilize the T structure of hemoglobin, lowering the affinity towards oxygen. So in tissues, oxygen is again released, and the opposite takes place in lungs. Okay, so let's talk about the other allosteric effector. But before that, let's talk about the oxygen level at different altitudes. So at sea level, the atmospheric pressure is about 760. Okay. Now arterial oxygen is about 100 torr. That's what exists in our lungs. Now in tissues, the pressure of oxygen is about 40. Okay, so there is reduction, or there is release of about 60 torr of oxygen in tissues. Okay, good. Now if you go high, if you go to a high mountain that has an altitude of 10,000 feet, which is about 3,000 meters, the atmospheric pressure goes down. Now arterial oxygen goes down as well. So it becomes 60. Okay. Now in tissues, the pressure of oxygen is about 30. So about 30 torrs of oxygen would be released, and this is really very little compared to normal conditions. That is at sea level. So of course, the higher that you go, the lower the oxygen pressure goes, and same thing with arterial oxygen and so on. Okay. So the major player is a molecule known as 2,3-bisphosphoglycerate. Now 2,3-bisphosphoglycerate is a byproduct. Now you learned in glycolysis, glucose is converted to pyruvate, and it goes through an intermediate known as 1,3-bisphosphoglycerate. Now 1,3-bisphosphoglycerate can be converted into an isomer, which is 2,3-bisphosphoglycerate. Okay, and it can go back, by the way, to the glycolytic pathway, but this is the main player right here. Now notice the number of negative charges that this molecule carries. Okay, now this is the major player. So what does it do exactly? Now 2,3-bisphosphoglycerate binds to hemoglobin at a ratio of 1:1, and specifically it binds inside the cavity of the hemoglobin molecule. That is exactly in the center of the hemoglobin molecule. Okay, and it binds mainly to hemoglobin in the T state, that is deoxyhemoglobin, and it forms a lot of electrostatic interactions with surrounding amino acid residues, including a lysine. So you have two lysines right here, left and right. You have histidines and so on. One of these histidines is histidine 143, and there is a reason why I mentioned this histidine particularly. Okay, so what happens is that you have a lot of electrostatic interactions that are formed, and these electrostatic interactions stabilize hemoglobin in the T state. So it's almost like the hemoglobin molecule wants to rotate. The polypeptides want to rotate around the axis, and bisphosphoglycerate blocks the rotation of the hemoglobin molecule from the T or converting the hemoglobin molecule from the T state to the R state. So what happens is that when oxygen binds to heme, that there is sort of like resistance in changing hemoglobin from T to R. Okay, eventually what happens is that the bisphosphoglycerate is kicked out. It gets released from the hemoglobin molecule, and the molecule, the protein changes from T to R state. And 2,3-bisphosphoglycerate, what it does is that it shifts the curve to the right again. So it reduces the affinity. So note here, you have different concentrations of 2,3-bisphosphoglycerate. Here you have none at all, and note that the sigmoidal curve right here, but the curve is shifted to the left. So this has very high affinity. So it almost looks like myoglobin, which has really high affinity, but it's not there, but it's close. Well, if you have in normal blood, you have 5 millimoles of bisphosphoglycerate per liter. So you have the normal saturation curve. But if 2,3-bisphosphoglycerate increases to 8, note that the curve shifts to the right, meaning that you have low affinity hemoglobin in this case. And it turns out that individuals that live in high altitudes, they adapt by increasing the amount of 2,3-bisphosphoglycerates in their cells. Okay, so what happens is that if you go to a place that is at high altitude, the first 2 days you would have a hard time breathing as you're walking, but on the third day and further, your body adapts by increasing the level of 2,3-bisphosphoglycerate in cells. So this is a case right here where, again, that's normal curve for hemoglobin, and here if you have no 2,3-bisphosphoglycerate at all, note the difference in the P50 and the shift of the curve to the left. Now, the effect of 2,3-bisphosphoglycerate in releasing oxygen at high altitudes, when a person is at high altitudes, can be explained a little better in this slide right here. So let's say that at sea level, about 100% of hemoglobin molecules are charged or saturated with oxygen. Now, in peripheral tissues where the level of oxygen is about 40 torr, let's say that about 55% of hemoglobin molecules are now saturated with oxygen. So you have a reduction from 100 to 55. So that means you have release of 45% of the oxygen molecules that are carried by the hemoglobin molecule. Now let's say that a person goes to a high altitude and there is no change in the level of 2,3-bisphosphoglycerate in red blood cells. Now the pressure of oxygen, now arterial oxygen, is not 100, it's about, let's say, 80 or let's say 75. Okay, let's say 80. Okay, so you have less hemoglobin molecules that are saturated with oxygen. Okay, now when it goes to tissues where the level of oxygen is about 40, now it goes from 80 to 55, so that's release of about 25% only. Okay, but when there's increased concentration or level of bisphosphoglycerate in red blood cells, the affinity or the curve shifts to the right. Okay, so that results in a new curve as shown in here. So when at high altitude, when the pressure of oxygen is about, let's say, 80, now yes, you have much less hemoglobin molecules being saturated with oxygen. Okay, let's say about 70%. But then when this hemoglobin reaches peripheral tissues, now the pressure of oxygen is about 40. Notice that let's say that it goes down, the level or the amount of saturated hemoglobin now is about 25%. So you have reduction from 70% to 25%. And this is a release of 45% of oxygen molecules from hemoglobins. Okay? An important point about 2,3-bisphosphoglycerate is that its level goes down when blood is stored. So if a person donates blood and this blood is stored even at 4 degrees, the level of 2,3-bisphosphoglycerate goes down within hours. So after 24 hours, there would be no 2,3-bisphosphoglycerate. Now, this shouldn't be a problem with normal individuals or people who are not severely ill, because they can restore the depleted levels of 2,3-bisphosphoglycerate within hours. But severely ill patients would be compromised because they would need 2,3-bisphosphoglycerate in their blood in order to successfully and easily release oxygen as hemoglobin gets into tissues. So for these patients, 2,3-bisphosphoglycerate would be added, as well as ATP would be added to samples, to these blood samples. Now, temperature also has an effect on affinity of hemoglobin towards oxygen. The higher the temperature, the lower the affinity. So what happens here is that, as you can see here, that's normal body temperature or almost normal body temperature. As it goes up, you can see that the curve shifts to the right. And that is a good idea. In a way, if you're exercising, for example, you want oxygen to be released easily in tissues. And if someone is sick as well, tissues must have high metabolic rate in order to fight the disease or whatever. So what happens is that increased temperature can also have an effect on the metabolic rate of red blood cells. That would increase the production of 2,3-bisphosphoglycerate, and that would facilitate oxygen unloading from or release from the hemoglobin molecule. So there is a dual effect or double effect of temperature on hemoglobin affinity. One is direct by affecting probably the structure or the interaction, the binding between oxygen and the iron of the heme molecule. And the other is that temperature would increase the metabolic rate of red blood cells. Now, there are other considerations as well. So let's talk about fetal hemoglobin now. Now, fetal hemoglobin has higher affinity to oxygen compared to adult hemoglobin. So the curve shifts to the left instead of the right, okay? Meaning that, again, the affinity is higher. You need less oxygen to fill 50% of the hemoglobin molecule, fetal hemoglobin molecule. And the reason why fetal hemoglobin has higher affinity is because histidine 143, which I've talked about before, is replaced in the beta subunit, is replaced by a serine in the gamma subunit. Now, this histidine 143, is important in forming electrostatic interactions with bis-phosphoglycerate. So when this histidine does not exist, it means that you have less electrostatic interactions, you have weaker interactions between bis-phosphoglycerate and the hemoglobin molecule, and as a result, the fetal hemoglobin has higher affinity towards oxygen. And this is really an advantage because the mother can compensate the low level of oxygen in her blood by breathing faster, let's say, okay, by taking deep breaths. Whereas the fetus has only, it has to act as a parasite. It has to extract or capture or steal the oxygen from the mother. And there is also an effect for carbon monoxide. Remember we said that carbon monoxide is toxic, right? It's fatal. And the reason why is that it has very high affinity for hemoglobin relative to oxygen. So that's oxygen saturation curve for hemoglobin, and this is the curve for carbon monoxide. You can see that carbon monoxide has much higher affinity. Well, it also has, it increases actually also the affinity of hemoglobin towards oxygen as well. So what happens is that whenever you have carbon monoxide bound to oxygen, the affinity is higher. Except that it's not a good idea because oxygen is not released. It keeps on binding to the hemoglobin molecule. So the saturation, so what happens is that not only that carbon monoxide occupies sites for oxygen binding, it also increases the affinity, preventing hemoglobin from unloading or releasing oxygen in tissues. And probably that's why it's toxic. So carbon monoxide can be fatal. Now the thing is, if the level of carbon monoxide in inspired air increases to about 1%, it can be fatal. Now, as a result of pollution due to factories, the concentration of carbon monoxide in blood can reach 1% in non-smokers. In smokers, it can get up to 10%. Now, if this concentration reaches to about 40%, as is caused by this 1% right here in air, it can be fatal to people. Now, these are the different chemical components that you can find in cigarettes, and they include things like things found in cleaners, candle wax, batteries, poisons, and so on, and I don't understand why people smoke. So, to sum up the lecture, we have the saturation curve for oxygen, okay? And it can shift to the right or to the left. If it goes to the left, affinity increases. If it goes to the right, affinity decreases. These are the different factors that affect affinity. Fetal hemoglobin, we talked about it, it increases, it has higher affinity as a result of this amino acid change from histidine to serine. Carbon monoxide also increases affinity, but it's fatal because it not only that it occupies oxygen binding sites, it also prevents the release of oxygen in tissues. These are the factors that shift saturation curve to the right, decreasing affinity, lower pH, the Bohr effect, the effect of CO2. We talked about two mechanisms, the effect of 2,3-bisphosphoglycerate, and we talked about temperature as well.